I'll let you know. Okay. You're on.
Good afternoon and welcome to the St. Croix River Association's Brown Bag Lunch Forums. My name is Don Hansen. I'm the chair of the Water Resource Committee for the River Association. Today we have the pleasure of having Dr. Mark Edlund, a senior scientist from the Science Museum of the Minnesota St. Croix Watershed Research Station with us. And he will be talking about the good, the bad, and the slimy in the St. Croix River. Look forward to hearing that. A little bit about Mark. Dr. Mark Edlund is a senior scientist at the Science Museum, located down by Marine on the St. Croix. Through the years of experience, including a Ph.D. in natural resources and environment from the University of Michigan, and his appointments as Adjunct Faculty with the University of Minnesota and the National University of Mongolia, his research interests have developed around aquatic biology and algae, especially diatoms. More specifically, an element of Dr. Edlund's current research is focused on the use of diatom subfossils to measure change in historical diatom communities, lake productivity, and nutrient levels in response to climate and land use changes and management practices. So, with that, welcome, Dr. Mark Edlund.
Thank you. All right, thanks for that introduction, Don. Yeah, I study algae because someone has to. There's not a lot of people out there work on algae. I work at this place called the St. Croix Watershed Research Station. I got the job there back in 2000. I walked in and they had the best microscopes I've ever seen in this area of the country, and they were looking for someone to work on algae there. And it was like, you know, a match made in heaven. So people who work on algae they stand out. You know why? What do you call them? What do you call someone who works on algae? Well, Roger Daltrey said, "Who are you?" And this is what someone who works on algae is called, a phycologist or an algalologist. But you know, we wear a lot of other hats. I think in general, I would call myself an aquatic biologist or a paleoecologist because I study past environments using algae. And my real specialty and real passion is one group of algae that are called diatoms. And so I also might call myself a diatomist in things. What I want to talk about today is algae in general. We'll talk a little bit about what are algae. I think people have some idea what algae are. We'll talk a little bit about the characteristics of them. And then we'll talk about what good algae are and what some of the bad algae are. And then what I want to finish up with is how some of the research that we're doing at the St. Croix River, or the St. Croix Watershed Research Station, is using algae to address some of these larger environmental issues and managing lakes and rivers in this region. So what are algae? Algae are actually a non-natural group. It's like if you use the word bugs. Bugs are creepy crawly things. You know, it might be a spider, it might be a bee, it might be an ant, it might be a centipede. But algae are the same sort of thing. They're just a group of organisms that we kind of put together. They're typically found in aquatic or marine environments. They're ubiquitous, found in most every place. They typically are photosynthetic, so they're taking energy from the sun, converting it to food at the base of the food web. From a sort of a scientific definition standpoint, they are non-vascular, so they're not plants. If we go really fancy, we start talking about them reproducing without a sterile layer of cells. So again, they're not flowering plants or ferns and things like that. They range in sizes from 1 micron, so that's 1,000th of a millimeter, up to things that are maybe 50 meters long, like the kelps. All these things are grouped in this very non-natural group called algae. They're very important from a question?
Well, before you get done with this slide, I would like you to give more definition to the non-natural. You know what I mean? It's like, in other words, they're not supposed to be here? Or
No. What it is it's a convenient group to say. I can say algae and people get an idea what algae are. But when we look, I think on my next slide here, ready?
Yeah.
Nope, one after this. You'll understand why they're not a natural group.
Okay.
They're everywhere though. I mean, you look, they're found in oceans, lakes, rivers, backwaters, floodplains, reservoirs, soil, and most are native. They've been in a region for a long time, and they're a common thing that's part of our ecosystems out there. Algae come in many major groups. Some of these things people have heard of. Most people have heard of blue-green algae or cyanobacteria. Most people have probably seen or heard of something called green algae. This is when you look at a rock on the side of a river or a lake and it's got green stuff hanging off it. That's usually a green alga that's going. Some things, here's me holding a kelp, a thing called Alaria. It's about 6 feet tall. That's a brown algae. The thing about this, there's probably somewhere 18 to 20 different groups of biological organisms that we cluster in this thing called algae, and many of these things we can find in the St. Croix River. But the key is that, and this is where I answer your question about being a non-natural group, is algae as a whole are found all over the tree of life. They're found in several different of the kingdoms, including the bacterial kingdoms, of things that we call algae. So that's what I mean when I say it's a non-natural group. They are not representing one evolutionary lineage. They represent, there's groups in the plant groups, there's groups in some of the animal groups, there's groups in the bacterial groups, and we just, out of convenience, call them algae. On the St. Croix River, we have all kinds of great places for algae to be and grow. We have an ecoregional gradient that goes from the boreal forests in the south to egg-dominated regions in the north. We have the pine forest, boreal forest. In the south, we have agricultural-dominated things. And of course, across this continuum of this river, we have things like backwaters and floodplains and reservoirs and springs. And in the terminal end of the river, we have Lake St. Croix, all great places to grow algae. And when we think about diversity of algae, I've studied, my specialty is a group called diatoms, and from the work I've done, I know there's at least 350 of diatoms in the St. Croix River, and I haven't studied them as hard as I probably should have. There's probably 3 to 4 times that many, actually, if I went and sampled every little habitat out there where these things could grow. Now, there's been some reports recently about green stuff in the river in the winter. And as always, I usually say, bring us a sample, let's take a look at it, see if we can figure out what this is. It's a little strange to see green stuff in the middle of winter. Normally the water's pretty nice in the St. Croix River in the winter. So yesterday we got a couple bottles of water, put them under the microscope, and here are the things that were in the St. Croix River yesterday. What color is that? Green? A little more brown, actually, right? So what we're seeing is different groups of algae. There's actually no green algae in this picture. Some things you guys have heard of is Euglena. People have probably heard that name. These things in the middle are diatoms. This is actually a blue-green alga here. It's called Waronikia. This is the chrysophytes here, dinoflagellates. So there's a whole bunch of different groups of algae that are right now living in the river. What I didn't see was anything that would have been causing this coloration of the river that people were talking about. I'm not sure what it is. It may be I don't know. I don't want to speculate until I see it. Algae are good. They form, in the aquatic food webs, they form the base of the food chain. They're taking energy from the sun and bring it to food, and when you do that, things eat you, and it goes right up the food chain. This is a chironomid, which is a little non-biting fly whose larvae lives in the river. If you squish him and look at his guts, he's full of diatom algae that he or she is eating. They're so important that they actually, algae as a group, do about 50% of the photosynthesis on the earth. That means that every other breath you take, you better thank an alga. Okay? That's how important they are. If we didn't have algae in this world, we'd have a sad, sad state. Unfair. Algae are moving right up the food web, or at least the energy that they've taken moves up the food chain. What do fish taste like? Fish, right? What do you think algae? What do you think algae tastes like?
Fish.
So what do fish taste like? Actually, yeah. The energy and some of the like fatty, the omega-3 fatty acids that are in fish are actually initially generated by the algae. So fish, yeah, fish tastes like algae because things go right up the food web. Other things that are good is when you have a child, you can convince them to actually be excited about algae. This is my daughter, and I can convince her to pick up algae and hold it and let me take her picture. But she actually, she might go down the route of being a scientist one day, but ever since she was a little kid, we get her to pick up algae and look at them. Here's a number of fun shots from the seashore, of course. Other things that are good about algae is there's efforts being put forth to use algae as biofuel sources. Some of the things that have been discovered by doing that is it's not the most efficient process to make biofuels. Instead, it's a great way to get some secondary metabolites that are used in food additives and things like that. A much more economical approach to doing that. They're used, sometimes along the seashore. They use algae for food for livestock, but they are, used for other interesting purposes too. Now, I think for most people, algae are something we see as something bad or slimy. It's because when you get enough of them growing in a system, they start accumulating and they cause problems for us as humans. Generally, when we think of algae as having too much, it's often caused by too many nutrients in a system. Algae are microscopic. They're not all microscopic, but they're plants, so they need to have nutrients, and they also need light and temperature, and things like this can control. And of course, they have impacts to our ecosystems as well. Algae affects the water clarity. This is a paper that came out earlier this week. The title of the paper says, "Recreational Demand for Clean Water." And here's the punchline of it. In Minnesota and Iowa, lake users are willing to travel 56 minutes farther for every 1 meter of increase in water clarity. So 56 minutes is about 50 miles. So people are willing to drive an extra 50 miles to go to a nice lake compared to a lake that is not clear. And of course, the reason that many of these lakes are not clear is because they have too much algae in them. And we get things like algal bloom advisories put in place. This was a neat study that was actually based on using photographs that were put on the Flickr website that were geo-referenced to specific sites. It's a real novel approach to doing that. It just came out a couple of days ago. But the reasons algae are, of course, part of the factors that describe what happens in lakes. We think of lakes as being described as what is called a trophic state. Many people have heard the word eutrophic or mesotrophic or oligotrophic that are often reflective of how much phosphorus is in a system. In Minnesota, in a lot of places, typically fresh waters are phosphorus limited. What that means is that if you add more phosphorus, you grow more algae in those waters. When you add more nutrients, you promote algae growth. It often changes the algae that are there, sometimes getting species that are less desirable. It impairs the water for drinking, navigation, swimming, wildlife, uses like livestock, things like that, and recreational things as well. And in the end, if things get too far down this scale of phosphorus system, we end up with nuisance algae in systems. So what do you call a nuisance algae? I think for me, a nuisance algae is when we see visible growths or accumulations of algae that, as a human catches our eye and we say, something's up here. A lot of times we see these in the late summer or fall in lakes and streams. They impact our enjoyment, recreation of the river. They affect the value of the property that you have on the river. Obviously, people are willing to drive more if there's fewer algae in the system. And they affect ecosystem services, which means they affect the ecology of the lake system. Probably the worst thing that can happen is that in in some cases, they actually produce toxins. We see every year in Minnesota a few dogs that get killed by drinking tainted water, especially out during the pheasant season. They go, they're hot, they find a body of water, they drink some water, they ingest these toxins and die. When I think of the St. Croix River, I think there's sort of 4 kind of nuisance types of algae that we encounter in the St. Croix River. The first is cyanobacterial blooms, blue-green algal blooms that just turn the water into this sort of pea-green soup. I know Richard Kiesling spoke a month and a half ago, whatever, on this subject specifically down at Lake St. Croix. I don't want to talk about everything that he probably spoke on, but in general, these blue-green algae are found in the summer and fall in lakes. We normally find them, we typically find them in sort of these middle productive and more productive lakes. They're often very, very common in shallow lake systems, and they have some great adaptations to do what they do. The first thing is they're N fixers, so they're the only algal group that's able to take nitrogen from the atmosphere and convert it into nitrogen that they can use and other things can use. So it's the same process that occurs on the root nodules of soybeans. Not done by algae and soybeans, of course, done by another bacteria. They can regulate buoyancy. So if the nutrients are down in the bottom waters, the blue-green algae can go, I need to go get them. During the evening, I'm going to go down, pick up some nutrients, then I'm going to go back up during the day and get some light, because I need nutrients and light to live. They do produce toxins. There's a number of species that produce toxins that are dangerous, and this is something that don't go swimming in this kind of situation. Don't let your kids in it, don't let your dogs in it, don't let your cattle in it. You might have problems down the line. From an ecological standpoint, they're also oftentimes unpalatable. So other organisms in the food web are not that interested in eating these things, which is another reason that they're successful. Here's a couple pictures of down on McKinney Beach on the Wisconsin side of the river 2 summers ago, and even in the by Log House Landing of blue-green algal blooms. There's just really a handful of species that sort of cause these problems. We always call them, when I learned about them, we called them Annie, Fanny, and Mike. And they refer to the names, the scientific names. Annie is a blue-green alga called Anabina. Fanny is one called a Phanozomenon, and Mike is Microcystis. And all 3 of these things are part of these typical bloom-forming cyanobacteria that are found in these blooms that are especially down in Lake St. Croix. Now, is this a new phenomenon for the river? No, it's not. Actually, we can look back at the literature, back in publications from the 30s, blue-green algal blooms were described and discussed as part of St. Croix River even back in the 1920s. They are definitely linked to nutrient loading in this system. We study sediment records and look at, in this case, I looked at fossil pigments, so blue-green algal pigments, and we look back in time at the bottom of the graph and going towards modern, we see that there were blue-green algae back then, but since about the 1960s, they've really ramped up in their abundance in Lake St. Croix. We also know from some of the efforts to fix the St. Croix, make the St. Croix better, that we should be able to control these. We should be able to tamp these blue-green algal blooms down if we start controlling things like nutrient loading to the system. And that's been a real major directive for management efforts on Lake St. Croix. From a monitoring standpoint, we know that Lake St. Croix, and this is work that Sue Magdalene at our research station has pulled together. Lake St. Croix is seasonally dominated by blue-green algae. The sort of robin's egg blue thing here is across each year, they're the dominant alga that's growing in the river. You go downstream, we also notice that towards the mouth at Prescott, that they even typically become even more abundant in the southern reaches of the river. But it differs by site and it differs by year as to how much of a problem the blue-green algae are. Other recent research, I know that Richard Kiesling spoke about some of the whole lake models that they're doing to identify predictability of blue-green algae. And Sue Magdalene at our research station is working to understand how the different flow regimes of the river, whether we have high flow years or low flow years, and whether they're contributing to the blue-green algal problems. These toxins that I mentioned, they have been found in the river. A master's student up at UMD studied the upper Lake St. Croix, or Upper St. Croix Lake, I think they call it, in the upper reaches of St. Croix, and found in 1 year many readings of microcystin above the World Health Organization's safe limits. This is not water to drink. This is not water to swim in. This is not water to be in, at least at that part of the year. A problem just for the St. Croix River? Absolutely not. This is a global problem. Some of you probably heard last year, the city of Toledo, Ohio had to shut down their water intakes in Lake Erie. This is what Lake Erie looked like from the air. This is what it looked just a little closer. This thing was growing algae, growing blue-green algae to a point where toxins were being detected in the water intakes in the city of Toledo. And they were drinking bottled water for a good chunk of the summer. We're working on Lake of the Woods. Can you see Lake of the Woods in this picture? It's actually down here when it's full of blue-green algae. Looks a lot like the forests that are surrounding it, the same color. It's chlorophyll. But we've also found that wilderness lakes, we've been working on lakes in Isle Royale, Voyageurs National Park, that strangely are throwing blue-green algal blooms. These are wilderness lakes where you need to hike 2-mile portage to get to them, and they have a blue-green algal bloom. There's nothing happening in the watershed to cause something like that. No trees being cut down, no wastewater treatment plant being built, nothing like that. And we're working on trying to understand this phenomenon.
Got any ideas?
We got ideas. Yeah. I mean, we put in some proposals to try to do it. I think it's a link between climate drivers and possibly some nitrogen. In Minnesota, like I said, every year we seem to be able to, kill a few hunting dogs every year. Some of the earliest reports of algae in Minnesota were based on cattle dying from blue-green algal toxins. And this back way in the 1800s when we were first settling there. So it's a global problem. You can see this every continent, maybe not Antarctica, almost every continent in the world is having issues with blue-green algae. What other kinds of bad things do we see with algae? Another blue-green thing that we often see are these things I'll call benthic mats. Benthic algae are things that grow on the bottom. In the St. Croix River, we seem to get these blue-green algae that grow on the bottom of the lake, and then during the day through photosynthesis or oxygen production, they start floating, or maybe it's due to motorboat traffic, and they start floating downriver, and we accumulate in these leeward side or backwater quiet areas, and they're kind of nasty. They look like yucky stuff in the water. We've been getting more and more kind of reports of these things, especially south of the Apple River mouth, but also seeing them up by Copus, and even in our backwater by our research station. Interesting, it's one species of alga that seems to be the thing. They're kind of beautiful when you put them under the microscope. They're kind of like, you can kind of play with them if you just look at them in a dish here. They can move. They crawl out of their little gob of stuff that's floating in the river, and look like these little hairy tufts here. It's a blue-green alga called Oscillatoria lamosa. Fortunately, it's not a toxin producer, and it's pretty common on benthic algal mats. It's just that we're sensing that we're seeing more of them than we had seen in the past. Another thing that we typically will see in the St. Croix River are, rather than that blue-green color, this more brightish green color, and this is actually green algal mats that appear. Again, they seem to be associated and coming out of some of these backwater areas and flowing into the main stem. And this was a big accumulation of them coming out of the backwater by our research station and accumulating in docks just immediately downstream on the main stem. Several different culprits. They seem to be sort of the greens seem to be able to do things a little earlier than the blue-greens do. And they form these big macroscopic or visual things. When we look at them again, algae are kind of pretty, you guys. They don't look so bad. Two main culprits in here. One is this thing called Dictyon reticulatum. It's one of the few algae that have a common name. It's called water net. And you can tell why. It forms these beautiful mesh networks. The other one is Spirogyra, which is it's a green alga that is not a jazz band from the 1970s and 80s. You may think it is, but the algae got the name first. When you look at them just in your hand, the water net looks sort of like this. Spirogyra looks like this sort of green stuff. And again, they look kind of nice under the microscope. The other type of algae, or nuisance reports we sometimes see in the St. Croix River, are diatom mats. Diatoms are another group of algae that are brownish colored. They're not green. They have a different set of pigments in them, which give them that golden brown color. But what we typically see are sort of these gelatinous gunks, either floating or stuck to almost everything on the bottom of the river. They seem like they'll just sort of cover everything, and we've seen them at all different times of the year, spring, summer, and fall. One of the things, the first time I was brought up was folks were diving at the mussel beds just downstream of here and seeing huge masses of these things here. Up by Nevers Dam, these things on the bottom have dislodged and we're now floating on the top. And they feel like big gobs of snot. Sorry, folks. Why do they feel like that? Because they're slimy. It's weird. When I look at them all, everyone I've looked at like this is actually primarily one species of diatom. It's called, again, things are too small to have common names. It's called Cimbella turgidula. It's a weird little thing. It seems like every year it's the same species. It's native. It's been here for a long time. And this sliminess is because they produce a huge amount of stock material. Each of these diatoms here actually lives on the end of a mucilage stalk. When you look at them, these are mostly all dead ones here, but when you look at live ones here, here's the live diatoms. Everything else that you see in here, if you can see on the picture, it looks sort of like a whole bunch of rice noodles. Those are diatom stalks. So that's really what you're seeing is much more stock material, this extracellular mucilage that they've extruded that accumulates. And it's resistant. It doesn't break down really easily. So that's why it lasts for a long time in the environment. But one of the reasons that these diatom accumulations have been coming to people's attention is that throughout a good deal of the world and in the US here, we're seeing an actual, I hesitate to use the word invasive, but a diatom that is doing things that it didn't do in the past. It's called Didymo. Didymo is the only diatom I know that has a common name. It's called rock snot, because that's what it does. When it becomes common and abundant in typically a stream system, this is what you get, these huge piles of diatom growth on everything. We've watched these things increase in abundance throughout Canada, the US, Iceland, some streams in Europe, and all over the US actually. Arkansas, the Northeast, Colorado. And it's really a strange thing. When I met this diatom for the first time, it was on Lake Superior, where it's native. It happily lives on the rocks along the shores of Lake Superior. But even there, we're sensing that we're seeing greater abundance of this thing right now. I told you it's common. It makes big news when it invades the system. The system, what ends up happening is the system ends up looking very different than it did the year before because of all that accumulation of what is called rock snot. In New Zealand, which is the poster child for how we should think about aquatic invasive species, they've taken this seriously. They've put out campaigns trying to prevent this thing. Most of the evidence suggests that this was introduced into New Zealand. Why is it introduced in New Zealand? Probably anglers. Probably because you can go fishing in Alaska, and a day and a half later you can be fishing in New Zealand with the same wet boots on. These are... Why should New Zealand care? Because it's cost them $158 million over the last 8 years. That's, you know, that's a big deal for something.
Other than, Mark, the disgusting nature of it, what are there other dangers to it? I mean, yeah.
I mean, danger is all, you know, when we think of danger, we think of like human impacts. So it's not poisonous.
Okay.
But if you make your money guiding trout streams, is that dangerous? Yeah, it is. You just don't have a job anymore. You're unemployed. I mean, that's a danger, right? But in, you know, they also have ecological impacts. So when this gets into a stream, it changes the ecology of the system. It changes, in particular, the aquatic invertebrate community, the insect communities in the streams, to a point where it then affects the fish communities in the streams. Of course, that's where often the money is. Again, there's clearly a human component to the spread in some places. Jury's out on whether this is just a diatom gone bad, or whether it's being introduced across many places in the U.S. It may have actually been there in very low abundances, and it's just taken off. Nice study that was done a few years ago, 2009, looked at risk factors and where this thing could show up, this rock snot. And fortunately, the St. Croix is actually it's not flashing red there. It's actually a low risk thing, where it seems to be controlled as climatic factors. It needs very low phosphorus streams, which the St. Croix is not terribly low in phosphorus, and it likes stable flows as well. So I you know, keep looking for things like this. When you see these round gobs of stuff in the St. Croix, we want to keep track of this stuff. But right now, at least the modelers say we're not at a high risk for getting this thing in the St. Croix.
Mark, do we know, can they live all year long here in this climate?
They're living in Lake Superior. So,
okay.
But again, Lake Superior is a very low phosphorus. What's strange about this diatom is that it is able to create these incredible gobs of biomass on very, very low nutrients. And that's really the sort of the management thing, that conundrum that drives us. We don't understand exactly how to do it. It actually is probably related to the fact that they grow in these huge clumps and actually are able to strip phosphorus out of the water by creating a microenvironment within their slime to do it. I mean, this is where the research is sitting right now, is trying to understand how these algae can grow that kind of biomass with that little nutrients. Let's talk a little bit about what we do with algae at the research station, putting them to work. And as I was putting this talk together, I thought, you know, really, what I do is I use dead algae to help us solve environmental problems. The dead algae is stuff that's preserved in the bottom of lake sediment. So we're using sediment cores from the bottom of the lake to understand how a lake has changed over time. This might give us pre-monitoring conditions. We don't know what the St. Croix River was like in 1940 because we forgot to measure it. We only started measuring it in the 1970s. So if you want to know what the St. Croix River was like in 1940, you got to find a record. And the sediment core can provide that record. We can go back even farther in time and identify when and why and how much a lake or system has changed over time. And that's been a real important facet of our work at the research station. Talk about this sediment at the bottom of a lake. Sediment accumulates in layers at the bottom of a lake in very conformable things. So this year's sediment is what's sitting right at the top, and as you go down deeper into the sediments, it's older mud. We can go back, depending on how much mud you take out of the bottom lake, you can go back tens of years to even thousands of years. Every lake in Minnesota has somewhere between about probably 10 and 75 feet of sediment that has accumulated in it since the glaciers left. The lakes of Minnesota are 13,500 to 11,000 years old, and that's how much sediment has accumulated in them. So we can sample that and understand how lakes have changed over time. What those sediments preserve is a bunch of clues that include what's happened in the lake, what's happened in the watershed, and actually what's happened in the world. And it's our job as scientists to interpret that by looking at biological, chemical, and physical things that are preserved in those sediments. We use various methods to rescue that mud from permanent burial. Primarily involves taking a tube and sticking it into the mud and pulling it out. If we go out on a big lake, we actually get oceanographic equipment to take these samples. One of the key things we have to do is figure out what the date each layer of sediment represents. We use a number of radioisotopes to do that. They're naturally occurring things, almost naturally occurring. Things like lead-210. One of them that's interesting is cesium-137, which was produced during the atomic bomb era, when we were touching off atomic bombs in the atmosphere, which seemed like a good idea at the time. We were actually putting cesium-137, a new isotope, all over the Earth. And when they stopped doing that with treaties with the Soviets in 1964, we stopped adding that. And typically in a lake sediment core, we see a peak of cesium-137 in a core that corresponds to 1964, and then it starts going back down again. But we use these different types of radioisotopes to identify levels in a core. So when we look at a sediment core, we know the top of the core is now, going down in time, back and back. And here at this gentleman's knees might be from 1650. Every lake, of course, accumulates sediments at a little different rates and stuff, but that's a fairly typical thing. We might find 1 meter to 2 meters of sediment that have accumulated in a lot of lakes in the last couple hundred years.
And generally, it looked like there was a 30-year difference in your.
I just put those in there. It actually, the sediment at the top of the core is kind of sloppy and runny and wet. And so this might be 2005, that might be 1990, and down here might be 1920 because there's less. It compacts, the sediment compacts together.
But you couldn't delineate it by year.
Oh yeah.
You can.
Yeah. And we have error bars on it. It might be 1910 plus or minus 5 years or 3 years, but we know that that's about 1910 right there. From the biological things we look at in the sediment cores, there's all kinds of stuff preserved in sediment cores. Some things are algae that are preserved there, but you can find fish bones and plant fragments and little zooplankton animal remains. These are sponges. You can find sponge spicules in there. This is the real friend that we use, are the diatoms. And it's true. You look at under the microscope, Lake St. Croix in the 1840s. This is what your microscope slide looked like. These are all diatom shells in here. There's a bunch of them in there. And if you go to current time, this is what it looks like. All right, look at that one real quick. Got it? Memorize it. Memorize it. Be the microscope.
Okay.
All right, what did you guys notice?
Some things got bigger.
Things got bigger? How about numbers? More? How about types of species?
Different.
See any little round ones in that one?
No.
See any little round ones in this one?
Yeah.
So abundance has changed, species have changed in response to what's happened in the St. Croix River over time. Diatoms are great. They're super abundant in sediments. So it's not like digging up a dinosaur and telling you what the world was like 100 million years ago. You know, I can look at, there's probably 50, there's probably 40 diatoms in that picture. There's not one dinosaur bone. So we're looking at huge numbers of things. A teaspoon of mud from the St. Croix River probably has, billions of diatoms in it, dead diatoms in it. And we can look at and analyze to get a picture of what's happened back in time. The diatoms are sensitive to change. I just showed you they're sensitive to change. You guys believed it when I showed you. We can identify them to species. That's You know, we can look at these under the microscope, a little bit of training, we can tell you exactly what species they are and understand how they're responding. They're well preserved. What these are little Coke bottles. So these diatoms surround themselves with a little glass shell that preserves most lakes perfectly beautifully in the bottom of the sediment record. So that we can go back 10, 100, thousands of years and see that diatom assemblages there. And one of the real keys that we've done is we've used them as a tool. People typically don't care too much about diatoms. What they care about is something like phosphorus, especially if you work for one of the agencies. You want to talk about phosphorus. We can look at an assemblage of diatoms under the microscope and use models that we've developed to estimate what the phosphorus level was. That means now I can tell you what the phosphorus level was like in the St. Croix River in 1940. I can tell you what it was in 70, I can tell you what it was like in 1850. And there was no one there measuring it back then. So and that's a powerful tool as you think about how you want to manage a system. How have we used these things? In the state of Minnesota, this type of approach, looking at sediment cores, has been used to develop phosphorus standards for Minnesota lakes. The US EPA says, you guys got to do this. Once you've set up these standards for phosphorus, if they don't meet it, they're impaired. If they're impaired, you got to come up with a plan to fix them. And for Minnesota, the PCA is the ones in control of setting those standards. They've used these same sediment core techniques, again, putting dead algae to work, to understand what the lakes in Minnesota were like and how they've changed. We were involved in this work, studied lakes all over the state of Minnesota. What we analyzed was the top of the core to look at what the modern condition was, and then we looked at the bottom of the core. We made sure we went back before Europeans arrived on this landscape, so we kind of knew the natural condition of these lakes and compared those. This graph has the natural condition of the lake in the blue stripes here, the current condition of the lakes in this sample size in the green. And you can look as you maybe go from the northern lakes and forests, which are way up in the Arrowhead region here, the lakes up there haven't changed much. But as we go downstate into our prairie and ag regions, the lakes have actually changed quite a bit. The other thing to note is that the lakes in the northern lakes and forests, even before Europeans arrived, are obviously very different than the lakes down in the prairie region. This is just the way Minnesota is set up. We have a gradient of lake types as we go through the state. But we can also see that down in the southern regions of the state, the lakes are very, very much impaired. So this one has twice as much phosphorus now in this set of lakes than it did in the past. What did this PCA do with this? They used this to set standards for the different lake types in the different ecoregions. So deep lakes have a standard, shallow lakes have a standard, depending on what ecoregion you're in. And these are the levels of phosphorus that they used. They didn't just use these data that we produced, but they also used some other models to provide a body of evidence to support these standards for our lakes in Minnesota. How about Lake St. Croix? This is a good story. This is good science really directing what we're doing on the river here. When I first started at the research station, we got involved in this project trying to understand this. Lake St. Croix, hey, it's nice, but has this river system changed over time? We know that when it flows into Mississippi, we get a big contrast with the Minnesota and Mississippi stuff flowing in compared to the St. Croix. We know these things are very different. And when you compare this to this, we think, oh yeah, Lake St. Croix is a great river. That's an awesome river. But has it always been awesome, or has it been better? Could it be better? And that's the sort of approach we took. We know that things have changed in the St. Croix. Cut down all the trees, right? And then where we could, we converted a lot of land to agriculture in the thing, especially towards the southern end of the basin. These are things that happened in the St. Croix Basin. Got a lot of people move in. It's a great place to live. Population has continued to grow and is expected to continue to grow in this basin. All of these things have affected what's happening in this river. Oh, back to the algae. What happens to the algae when you do all these things to a river? Well, what we find is that they change dramatically. That's what that slide I showed you was like. We go back to the 1850s, all of the diatoms are a type that typically, not all of the diatoms, a lot of the diatoms are a type that typically is living attached to bottoms that are reflecting clear water that probably has lower nutrients to it. So we move up in time, we start actually growing water in the water column itself. And that's typically a reflection of nutrient additions to these systems. A dramatic switch in how this river has functioned over time. If you monitored it in 1970, what do you get? That's when you start monitoring it. What's the river like? You miss all this, right? We never figured out what the river actually could be like, or what it used to be like. If we start monitoring right then, and just monitor here, we don't see those changes as dramatically. So we know this river used to be different, had different characteristics. I told you we could look at these diatoms and these sediment cores and estimate the amount of phosphorus in the system, and that's what we did. We look at the modern time and we see that phosphorus is up in the 40s and 50s and 60s, which is about 2.5 times higher than it was before Europeans settled. So we've added a lot of nutrients to the system. We're changing the algae, we're growing more algae. When we look at, I showed this graph earlier about blue-green algae. As we increase those nutrients, we've actually started getting these blue-green algal blooms that are in the river. And this study that we worked on here was really one of the key pieces of information that we used to direct the current management efforts that are going on in the St. Croix River. And really, all kinds of stuff changed in this river basin, not just the algae. We know population increase. We know that actually wastewater treatment plants came online and have been putting nutrients into the system. All of these things have changed, and this study really was critical to helping us direct our goals as to what we wanted to do in the river. We knew that the phosphorus concentration had increased over time and is too high because the river was declared impaired, and we need to set a goal. And the recommended goal was we're not going to send it back to pre-European times, but let's see if we can get back to the 40s. So the standards that were set were standards that were condition that the river was in back in the 1940s. In the end, this is what really, drove the science. And a few years ago, the state of Wisconsin and the state of Minnesota agreed to start dealing with this issue, and the river was actually declared impaired for nutrients. And this is where a lot of the progress now is being directed on the river is getting this system to meet these new goals that have been set for the river. What can we do about these algae? For us, we've got some projects, and I know that other groups are also dealing with these issues, like the USGS, trying to develop an understanding of what's happening in the river right now. The project that we have is going to be focused on these backwater areas, in particular dealing with these benthic algal mats that are appearing in the river. We have no idea why they're there, if it's really an issue, when it happens, why it happens. Our first study is going to be directed at where, what, when, and why these things are growing there and what happens. We're going to plan a year of some big surveys along the river from Taylor's Falls down to Stillwater to look at these issues. We're hoping to partner with the St. Croix River Association with some citizen scientists out there and have generated a little bit of initial data to try to understand what's happening. But we want people with their eyes out there looking for these problems that are out there and letting us know when they happen so that we can actually study them. Finally, we know that algae can be a nuisance, but we also know that they're an absolute critical part and natural part of any kind of aquatic ecosystem. This is something that happened out in our backwater by a research station a few years back. I don't know if you can see this red stripe that goes down the thing here, down the backwater there in that picture. Well, that's, I've told people it's probably blood in the water, but it's not. We looked at it, we collected some stuff, and actually it's one algae that has this red pigment in it that likes to sit up on the surface of the water. It's called Euglena sanguinea. When I was a graduate student at Michigan, we got a report from the cemetery right next door to the campus, and they had this in their little pond on the cemetery, and they were concerned that the graves were leaking. And no, it's an alga that's just capitalizing on some great conditions on that particular day or that particular week to grow enough that we can actually notice them, even though they're microscopic. There's so many of them that we can notice them. But are we seeing more algae in the river? We know we're seeing more than have been in the past. Right now, the recent trends seem we're picking those apart with some of these modeling and monitoring efforts in the river, trying to understand what's happening with those. When you see these algae, what should you do about them? If you're dealing with big problems and concerns, of course, the Minnesota Pollution Control Agency and the Wisconsin DNR are your go-to people. They're the water quality authorities out there. Wisconsin has a reporting mechanism in their website. But what we've noticed is a lot of people hear these things and they say, can I drop a sample off? I'm looking at this, and we're happy to take those at the research station and get a sense of what's happening out in the river right now. That's how I happened to get those pictures of what the river was like yesterday. We had a sample dropped off. But you can, I call you a scumbagger if you're picking up pond scum. That's a scumbagger. Be proud of that. And really, remember that, celebrate diversity out there. There's a lot of cool stuff about algae. There's nothing bad about them. I mean, they're bad. They can be bad at sometimes, but they're also really cool biological organisms out there that just a few of us get paid to study.
What's your last question there? How to spot a bad algae?
The crazy thing is, it almost becomes common sense. When you look at those lakes and streams, and they are green, and just that blue-green painted, the water looks horrific. Stay out of them. For humans, typically we're not going to sit there like a dog and drink a bunch of water just because we're thirsty in the lake, but they can cause dermatitis and issues like that. Different people have different sensitivities to it, but you can, drink this stuff and run into big problems.
So Mark, the same way, what I've noticed right down at Log House Landing is like almost every year, beginning with August, there's all those little green things in there. How long does that last? Is that the point where you just tell people, don't go in the river at all?
Common sense is a wonderful, wonderful thing that we as humans have, and why risk it? Go swimming in a different place, but they'll last. Depending on what conditions are in the river, like they're not thinking too much about it. They're just capitalizing on resources.
Sure.
They're out there, whether they have enough nutrients, have enough light, physical things, whether the river's in flood stage or low flow stage, are going to help drive what's happening to these things.
That's good to know. I'm not into swimming that much. I would be on top of the river and stuff.
And one thing to remember, and I should have put a picture in here, is that everyone should know what duckweed is. Duckweed is not algae. Duckweed is actually a flowering plant. It's actually, you get a cool award if you actually see it flower. But it is not the kind of problem. But often people think, oh, it's covered with algae. No, it's duckweed. A whole different thing.
Does anybody actually harvest any of this algae just for no other purpose than it's so ugly? Some of it.
I think in general it'd be a losing battle to try to do that. There are certain places. So before the talk here, we were talking about when you go to health food stores and have spirulina pills and things like that. Those are actually harvested typically from natural systems that are extremely productive. I'll let you make the choice whether to take those.
I heard that was being done on the apple over by Ingrid.
That they were
harvesting.
I've not heard that. Were they?
To use it?
No, just to get it on.
Oh.
That seems like
You can. I don't know how much effort that would be. You're oftentimes dealing with organisms that have growth rates of 1 division per day, or more, or faster, I should say. So you may, you take them out, and 3 you leave are already growing again tomorrow. So I don't know. I'm not sure if that would be.
Mark, I know you don't know for sure what's causing this But you said you think one is some climate change things, and the other one is nitrogen. Where's the nitrogen coming from? Is there more nitrogen?
Atmospheric. So we've really coated the Earth with nitrogen through atmospheric deposition, and it's deposited both directly on the waterways, but also deposited on our lands. And right now, there's some suggestion that interactions between climate and the land is enhancing, now moving some of that nitrogen into waterways and maybe rivers. But there's also lots of other suggestions that a warming climate enhances this. And it's based on how lakes are behaving in those warming situations. Do they stratify more? Do they stratify less? And how the algae are responding to them. Randy?
This is probably a dumb question.
Never a dumb question.
You keep a bird bath next to a patio, and within a couple of weeks, it's full of algae.
Yep.
Is it windblown spores or is it carried by the birds?
There are algae that are moving around in the atmosphere. So a lot of algae have resistant structures that allow them to dry out, blow around. My guess is that as much as you're trying to clean that bird bath, that you don't get everything, and so you just have a little inoculum in there. And it's kind of cool. There's actually algae that are very characteristic of bird baths. They like that, and it doesn't help that the birds forget and sometimes go to the bathroom in your bird bath as well to give it a little shot of nutrients in it. But there are sometimes, especially in the summer, you'll find a reddish color in your bird bath. It's from one of the. It's actually a green alga that's red. Any other questions?
Well, thank you, Mark.
Thanks.
Well, that concludes our Brown Bag Lunch forum for today. Next month, March 5th is our next one, and we'll be talking about the history of navigation on the river. And we'll have a presentation by Jean Sheffy-Anderson from the St. Croix Scenic Riverway. So please join us for that, and thank you all for coming.
Thank you. Thank you, Mark.